Good day, everybody. We are ready to start. We have two speakers for today, Amadeo Caflisch and Ioana Ilie. The talks, as usual, will be 30 minutes, followed by a Q&A session where you can post your questions. And at the end, we will have a joint session where we will let you in the panel to personally ask the speakers. And thank you both for coming. And Danilo, I'm passing it to you.
Okay, thank you, Magda. Good afternoon and welcome everybody to today's session of the Amyloid Symposium series. It is my great honor and pleasure to introduce the speakers of today, Amadeo Caflisch, which in turn will introduce his postdoc, Dr. Ioana Ilie. Amadeo received his doctorate degree from the ETH of Zurich and subsequently moved to the laboratory of Martin Karplus at Harvard University, where he deepened his interest in computational methods focusing on protein fold instability and aggregation. Then Amadeo joined the Department of Biochemistry at the University of Zurich, where he was later appointed as full professor of computational structural biology. He is now director of the Department of Biochemistry of the same university. Some of his many scientific achievements represent fundamental contributions to the development of computational chemistry. To make a long story short, I would just mention that he belongs to the development team of CHARM, and this group has developed and coded in CHARM the implicit treatment of aqueous solvent and showed for the first time that side chain interactions are crucial for amyloid self-assembly. Amadeo has also developed many simulation protocols for drug design and for fragment-based high-throughput docking. I will let the stage to him that will introduce postdoc Dr. Ioana that will start with his talk. Please, Amadeo.
Thank you, Danilo, for the kind introduction. It's a pleasure to be here, and I would like also to thank Professor Amamorti for the invitation. Ioana studied physics in Romania, and afterwards she moved to the Netherlands for a PhD in Twente. During her PhD career, she developed coarse-grained models and simulation protocols to investigate aggregation with a main focus at that time on amylin, on the Parkinson's Parkinson's disease. Now, afterwards, she moved to my group as a postdoc about 4 years ago, and she kept working on simulations of amyloid aggregates, but also other interesting biological systems. And she was offered a junior professorship position in Amsterdam a few months ago, and she will start there, I guess, tomorrow. So, Ioana, it's yours.
Thank you very much, Amadeo. Thank you for the nice introduction. And thank you very much also for the opportunity to present our work here. This is work that I carried out in the Caflisch group over the past years, and I'm really excited to share this with you today. Now, to be more precise, today I will discuss about the effects of antibody binding on the dynamics of the cellular prion protein. Now, prion diseases, they are neurodegenerative disorders, and in humans, they are known as, it's known as Creutzfeldt-Jakob's disease, and in bovines, the more popular term is mad cow disease. Now, prion diseases, like many of the other neurodegenerative diseases, they are linked to the misfolding and aggregation of a specific protein or peptide. In this case, we are dealing with the transformation of the cellular prion protein, also known as PRPC, into an insoluble isoform known as scrapie, PRPSC, which is known to be, or was shown to be, highly toxic. Now, structurally, PRPC consists of mainly alpha helices, so these 3 alpha helices that are highlighted in the ribbon representation, and very small beta sheet. It's also highly soluble and membrane attached. Now, the toxic isoform, scrapie, is known to be insoluble and is associated with mainly beta-rich structures. Also, PRPSC has a high aggregation propensity, and it has this ability to interact with the healthy protein, superimpose its, let's call it toxic conformation, and aggregate into higher order structures known as amyloid fibrils. PRPC, as I mentioned, is a cell surface glycoprotein, meaning that it is membrane attached. And it has been shown to be involved in myelin homeostasis. Now, structurally, PRPC consists of 231 residues, out of which the first 22 are cleaved during trafficking. To attach it to the membrane, the last residues are replaced here by a GPI anchor. PRPC consists of 2 really main parts. First, the first 100 residues that consist the flexible tail, which is intrinsically disordered. It's characterized by the fact that it has 2 positively charged clusters that I highlight here in green. And these clusters are flanking an octa-repeat region. So we have 5 imperfect repeats at the center of the flexible tail. The second part, that is also the really structured part, the globular domain, is the one that consists of these 3 alpha helices highlighted by the red rectangles and the small beta sheet. 2 of these helices are stabilized by a disulfide bond. Now, proposed therapeutic approaches to avoid the toxic conformation, so to avoid the transformation of PRPC into the scrapie isoform, have been developed throughout time. And one of these proposed therapeutic approaches is trying to avoid the transformation into these amyloid fibrils by designing antibodies that stabilize, that attach to the prion protein and stabilize its native structure. Now, a family of such antibodies have been developed at the University of Zurich in the Aguzzi group, and they are known as POM antibodies. So, for instance, if we take here highlighted in red, POM1 finds its epitope at the alpha-1, alpha-3 region, and it has been shown to perpetuate rapid toxicity. At the opposite pole, we have POM2, which actually binds at the octa-repeat region, so in the tail, and it was shown to prevent POM1-induced toxicity. POM6, another one of these monoclonal antibodies, binds similar epitopes to POM1. So the arrows show actually the overlap in the epitopes, and was shown to be neuroprotective against POM1-induced toxicity. As a matter of fact, also combinations of these antibodies, so coupling POM2 to POM1 was shown to have beneficial effects and prevent toxicity. Over the years, so in the past years, actually either the solid state NMR structures or the crystal structures of these complexes between the antibody and the prion protein have been resolved. And there are really still a number of questions that remain unanswered. And what we are directing our attention today towards is how the binding of these antibodies actually affect PRPC structurally and dynamically in this sense. Now for this, we actually perform molecular dynamic simulations of several of these systems. So first of all, we have just the globular domain of mouse PRPC. We have the complexes, so the X-ray structures of the complexes of PRPC with POM1 and POM6. We performed explicit solvent simulations under physiological conditions, and we simulated each system for accumulated 5 microseconds. So basically, we had 10 independent simulations that we evolved over 500 nanoseconds. Very important to mention is the fact that for these simulations, we used only the variable domain of the antibodies, and this is mainly because it's computationally very expensive to simulate also the entire antibody to simulate the entire antibody. Now, what we found was really quite surprising. Our first finding indicates that the antibody binding increases the flexibility of the cellular prion protein. So we plotted the root mean square fluctuations of the C alpha atoms along the sequence of the globular domain. We found that overall, so generally, the antibodies increase the flexibility of the cellular prion protein with predominance in the region around here, alpha 2. This is also visible. So I mapped actually the RMSF on the ribbon representation of the protein. And these increased fluctuations are highlighted by the bit lighter colors and the green colors in the complexes with the antibodies. A very interesting finding was actually the fact that the two antibodies have a different effect in the region of the epitope. So essentially in this methionine 138 up to almost to the arginine, the attachment of the POM6 antibody leads to increased fluctuations in this region as compared to POM1, which has the opposite effect. Now we were quite surprised about this finding, and we dug deeper, and we managed to correlate this increased flexibility of the cellular prion protein actually with a diversified network of contacts, of intramolecular contacts within in the prion protein. So basically, we found that antibody binding alters the network of polar contacts in PRPC. I will not go into the individual details of these contacts, but we actually managed to map all these contacts and to show them by performing a Sapphire analysis. And just briefly, this Sapphire analysis and the Sapphire plot was developed a few years ago in the Caflisch group. And what it does, it essentially helps us visualize the different states in our system. So essentially, we have a profile that is shown here in black along a variable. This is called a progress index, and it groups together snapshots based on their geometrical similarity. The Sapphire plot is also annotated with a kinetic trace. So if we look here at the top, we have these rows of black, red, and green dots, which correspond to simulations coming from either PRPC alone or in complexes with the two antibodies. Additionally, we used distances, so key distances that were identified either previously in the crystal structures or simply by visual inspection that were either formed through the simulations or were destroyed by the attachment of the antibody. So just as to picture it, if you have this arginine aspartate distance at the bottom, dark colors means that the two residues are really close, like highlighted here at the top. And as we go towards green, larger distances are sampled. Importantly, if we check the individual basin, we can see in which system that particular state is being populated. So take, for example, the basin over here, which is abundantly populated in the mouse PRPC system, that is in the free state, and also in the complex with the POM6 antibody. However, this is really marginally populated in the POM1 system. Now, just as an example, I focused here on these a network of contact that consists of the arginine 156, this aspartate anti-asparagine, and also the glutamic acid is taken into account. Now, in the free state, the arginine is free to move inside its neighboring pocket and interact with these three residues almost equally. So it can rotate and then form closer contacts to the glutamic acid. Now, for instance, if at the moment that POM6 is attached, residues from the antibodies are inserted. So in this case, the serine 32 from the light chain of POM6 inserts itself into close to this pocket of residues and favors closer contacts really within the pocket, which eventually leads to a decrease of interactions with the glutamic acid. Of course, there are several of these distances that play a role, but I want to convey a broader picture of this system. So I mentioned at the beginning that this protein has really a large tail and a long tail, but in the simulations I just introduced to you, we didn't take these tails into account. So the question is, what role does the tail play, and how can we actually capture the different states that the tail is sampling? Now, another point is the attachment of the prion protein to the membrane, which we didn't take into account previously. Additionally, in experimental hypothesis, it was proposed that POM1 docking and the toxicity associated with the attachment of POM1 to PRPC actually can emulate the binding of PRPSC, and this whole process is mediated by the flexible tail. So essentially what we wanted to achieve is to have a protein that is attached to a membrane here at the bottom, and has its full length flexible tail, so the 100 intrinsically disordered residues, and that should technically be free to move and sample all the possible states. Now, by explicit solvent simulations, this is really hard to achieve because we are dealing with really large systems, and not just the protein itself, but also the membrane is a very complex entity. To solve this problem, we went one step further and we performed, first of all, implicit solvent simulations using the absent implicit solvent model and explicit ions to maintain the physiological conditions. And we used enhanced sampling. So basically we used a combination of Monte Carlo and torsional MD and combined this with replica exchange across 24 replicas. So essentially what we do is a bunch of Monte Carlo steps and a bunch of MD steps in the torsional space. Additionally, to mimic the attachment of the protein to the membrane, we actually apply the harmonic potential, which allows here the last residue that just highlighted it in a sphere to mimic the GPI anchor, and it just bounces back and forth. Another aspect is the membrane, and the membrane itself is very complex. And we use a coarse grained representation of a membrane that we actually develop, in which we represent just the membrane as 100 acetate anions. that move along a flat surface. So basically, these represent just the lipid head groups, the negatively charged lipid head groups. And we performed the simulations for all the systems that I introduced before. So mouse PRPC in the free state, unliganded, and in complexes with the two antibodies. So the first step we had to do was to actually reconstruct the prion protein. And in this sense, we reconstructed all the 100 residues in the tail, and we relaxed them such that they come to as close as possible to a native structure. We applied similar protocols also for the complexes with the two antibodies and performed our implicit solvent simulations. Now, as a first analysis, we looked at the interactions with the membrane. And for this, we actually plot the distance of the C alpha atoms in the composition of our prion protein to the membrane layer. Now we have a very, I highlight here in the plot, so the secondary structure assignment, so the 3 helices by the red rectangles and the beta sheet over here. In the flexible tail, I highlight again the charged clusters, and by these dashed gray lines, I separate the individual octarepeats from each other. The red dashed lines, they actually highlight the distance, the POM1 epitope. Now we see a rather heterogeneous behavior of these distances, of the distance of the prion protein to the membrane, but we can't say much about it unless we talk about it in context also of POM1. So essentially, what we can identify by comparing these two plots is that the profile, the distance profile, actually changes dramatically. Now I will start from the end. In both cases, we can clearly see that the protein remains neatly attached to the membrane surface. However, the disposition of the helices is quite different, as in the wild type of protein, it seems that the helices and the globular domain is attaining a more upwards conformation. Here in the complex with POM1, it appears that this alpha-2 attains a parallel conformation to the membrane, and we shall see further on why. Additionally, we can clearly see that the antibody appears to, actually it reduces the distance of the globular domain to the membrane by almost 2 nanometers. And not only that, but it also reduces the distance of the flexible tail to the membrane. And in particular, we highlight here closer distances of these charged clusters, or the positively charged clusters to the membrane. And now what POM6 does, it has a quite similar effect on the proximity of the prion protein to the membrane. Here I highlighted the POM6 epitope by the purple lines, and you see the really clear overlap of the two epitopes. So overall, the antibodies, they actually push these proteins closer to the membrane. But what other effect might this have on the orientation of the globular domain with respect to the membrane? As I mentioned, we saw that alpha-2 can have different orientations depending on the system. Now, to study these, let's call it orientational flexibility of the globular domain, we plot the angles formed by the individual helices with the normal to the membrane surface. Now, the easiest to explain is for alpha-3. So essentially, we construct a vector connecting the end of this helix, and then we just calculate the angle to the z-axis. What we found was quite surprising. So if we focus here on the black lines for just unliganded PRPC, we find broad distributions for all the angles, indicating a higher orientational flexibility of the globular domain. And as a matter of fact, if we focus on alpha 3, and I overlapped several snapshots from our simulations, we can actually see that the protein appears to be quite mobile on the surface of the membrane. The behavior is, however, different when it comes to the two antibodies, because the distributions are not so broad anymore. They are narrower, which indicates more a localized and a restricted motion of the prion protein. And in fact, for both POM1 and POM6, we see that the prion protein lies flatter on the surface of the membrane. Yet they have a slightly different behavior. And there's no better way to explain this than with a movie. So for POM1, we see that throughout the simulation, so these are the unrexed results, the protein is really bouncing back and forth. I will play the movie again. Is bouncing back and forth, and it's quite dynamic. The tail is not stuck. In the complex with POM1, this behaves rather different. So the protein is still quite mobile in the sense that it rotates along on the surface, on the XY surface. The tail is also very dynamic, but we can clearly see that it never attains this upward conformation and it has a reduced dynamics as compared to PRBC. Now, similarly for POM6, we see that the protein has more of this rotating motion along alpha 3 and also with a dynamic tail. So we essentially characterized the interaction of the protein with the membrane and the effects that these antibodies have. Yeah, I'm running the movie again. Now the question is, what effect do these antibodies have on the interactions of the flexible tail with the globular domain? And for this, I performed a contact map analysis, which I mapped on the ribbon representation of the globular domain of the prion protein. On the left, we see a contact map analysis. On Y is the flexible tail, and on X, I apologize for this, is the globular domain, but it will become clearer in my next animation. And with the vertical dashed lines, I highlight the location of the POM1 epitope. What we can identify is that essentially the flexible tail interacts with very specific regions on the surface of the prion protein. So especially in the over here, which is the beta sheet region. So we can identify it by these first two stripes in our plot. And also with the C terminus of alpha 2, highlighted here by the darker, thicker colors, and which is also visible in the contact map. Now, we wanted to test the effect of the antibodies on these interactions. And for this, we calculated the difference map. So the difference between the contact map in the complex with POM1. From this, we subtracted the wild type, so the PRPC contact map. And now what we identified was quite interesting. So essentially, these interaction hotspots are preserved. So the flexible tail still interacts with the beta sheet region and with the C terminus of alpha 2, but the frequency of contact is altered. So to be more precise, we found that more frequent contacts are formed with the beta sheet, and these ones at the alpha 2 are clearly reduced. Similarly, POM6 has a different effect. So what what we saw was essentially an extension of these contacts. So besides just preserving the interaction hotspots, we saw that there are also occasional contacts that are formed between the flexible tail and here the N-terminus of alpha-1. Additionally, more frequent contacts are formed with the C-terminus of alpha-2. And yeah, in the beta sheet regions, the contacts are also slightly altered as compared to wild type. Now, this actually brings me to my conclusions today. So what we strive to do is to actually characterize the effect of these antibodies on PRPC. And we did this by both explicit solvent simulations as well as implicit solvent simulations. The explicit solvent simulations enabled us to characterize the flexibility of PRPC, which can be ascribed to an altered network of native contacts. I showed you just an example in which the antibodies are 6 pockets, and POM6 has an even higher effect. We then asked ourselves what the effect is of the tail and of the antibodies to membrane-bound PRPC, and for this we used implicit solvent simulation and the novel coarse representation of the membrane. We found that in both cases, PRPC is pushed closer to the membrane, so both the globular domain and the flexible tail, and that the antibodies restrict the orientational disorder of the globular domain. Additionally, these antibodies modulate the interaction frequencies between the flexible tail and the globular domain. Now, just to wrap up, this brings me actually to a point that I brought up on one of my first slides, and that was that the effect of POM1 on PRPC is to emulate the binding of scrapie, which transforms the protein into its toxic isoform, so it makes it beta rich, and all of this is mediated by the flexible tail. Now, while our simulations can't inform on the toxicity of the POM1 antibody, what our simulations actually support is the idea and underline the role of the flexible tail. And of course, further investigations are required. So for instance, whether there are any structural conversions in the globular domain, and also one cannot exclude the aggregation hypothesis, so the attachment and the grouping of several proteins on a membrane surface. So what is next in store for us? As you heard in the introduction, I'm actually starting my group at the University of Amsterdam, but we plan to actually continue in these directions of neurodegenerative disorder. And actually motivated by the studies that I just presented to you, we thought to use a different approach to block the toxic transformation of the healthy protein into these pathological prion protein. And we plan to achieve this by designing cyclic peptides, starting from everything that we learned from the previous study. So we will rationally design these peptides and test them in a computer simulations. And of course, we plan to validate them with experimental collaborations. So in this sense, we are actually looking for a collaborator that is interested in working together with us to test the binding energies, free energies of these peptides to the prion protein. And additionally, with this, I also want to hire a postdoc to work on this project. So don't forget to check our website at the University of Amsterdam. And now I come maybe to the most important part. So I cannot underline this more. I did this work at the University of Zurich, and I am very grateful to Professor Caflisch, who mentored me over the past years. I also want to thank everyone that I worked together on this project, so Marco and Andreas in the Caflisch group, and of course, the close relationship that we had with the Aguzzi group and with Assunta from the University Hospital in Zurich. Also, I want to thank my funding agency who made this possible, so the Peter und Traudl Engelhorn Foundation, and of course, I want to thank you for your attention. I'll happily take any questions, and I understood there at the end. Did I lose everyone?
No, not me.
Oh, okay. I thought I lost everyone. So I don't know how we do it now. I understood the questions are really at the end of the both of them.
I will share my presentation.
Okay, I'll
Okay,
stop
we can
them.
answer. We can read the questions at the end. So please, Amadeo, you can start your presentation.
So I will present work carried out in my group over the past 2 decades, and in particular, 2 main topics, the kinetic control of fibril formation and the design of antiprion compounds. So we are at the University of Zurich. In this campus here, this is the city, the lake, and this church is this one here. So it's a very beautiful place, and the campus is very close to downtown. We start with the simulation models. It is always important to have the right model for the question to be answered. And here we wanted to investigate the kinetics of aggregation. And Riccardo Pellarin, a PhD student in my group, developed a simple, coarse-grained phenomenological model. The model consists of a few bits, 10 bits, 6 large bits and 4 small bits, and has 2 main conformations, a so-called pi or protected conformation, which does not allow it to self-assembly. And then there is a so-called beta prone conformation in which the 2 dipoles, so the only 2 macrodipoles of the model, allow it to form ordered aggregates, fibril-like ordered aggregates. So the beauty of this very simple model is that because it's very much coarse-grained, one can use very large time steps. We were using 50 femtoseconds at that time. And one can reach high, or at that time we could reach high microseconds timescales. Now, as Ioana has shown, we have also worked with atomistic model, and as mentioned by Danilo in the introduction, Jörg Sponer used atomistic simulations already nearly 20 years ago to demonstrate the importance of side chains. These were simulations of a peptide from a fungal prion-like protein, and we saw spontaneous formation of parallel beta sheets, anti-parallel or mixed beta sheets, and we could investigate the free energy profiles of aggregation. Of course, this is slower because here it's 1 bit per atom, and the time step is 2 femtoseconds. So it takes much longer to reach microsecond time scales. But despite that, with a small system, so only 3 peptides, we could provide a strong evidence of the importance of the side chains, like in this case, the stacking of tyrosine side chains. At that time, there was essentially no structure known on amyloid-like aggregates. There was very little information. So again, 2 different models, coarse-grained for reaching longer timescales and much larger self-assemblies, and atomistic to somehow simulate the details of the side chains. And I would like to continue with the coarse-grained phenomenological model that with which we discovered the kinetic control. And the movie of what happens when one mixes in a box 125 of these monomers, there is a spontaneous formation of micelle-like structures or spherical oligomers, and also formation of protofibrils. And you can also appreciate that the density of isolated monomers diminishes substantially. And towards the end, there is only one main fibrillar state, though several metastable states have been reached. And when Ricardo investigated the details of the metastable states, he found not only the micellar oligomers, as you saw in the movie at the beginning, but also so-called protofibril consisting of 2 protofilaments or 3 protofilaments, with a number of monomers reaching between 50 to 70 or 60 to 80. And then there were certain species that were more rare. And one of the final aggregates was the 4 protofilament fibril with more than 90 monomers and 4 protofilaments, which you can see here in this picture. And actually, there were different morphologies of the 4 protofilament fibril. This one had two protofilaments going down and 2 up. This one had 3 and 1. And this one had also 3 and 1, but the structure was quite different because this one was a bit fatter. So we saw really a heterogeneity of fibrillar states, despite the simplicity of the model, which had essentially only one main degree of freedom, and the interconversion between beta protected and beta prone states. With the same model, Ran Friedman and other postdocs in the group simulated the aggregation of a fibril on top of a lipid vesicle, and we investigated the leakage of particles from the lipid vesicles, which emulates a mini cell. The interesting thing we discovered with RAN was that leakage was promoted by the growing of the aggregates and not by the fibrils themselves. As a matter of fact, when we ran simulations with the already aggregated fibril, we didn't see any leakage. So again, here you saw the growing of a fibril on the surface of a vesicle simulated with a phenomenological, very simple coarse-grained model. And when we then compared with experimental data, mainly electron microscopy images, we could appreciate, at least on a semi-quantitative level, that somehow the species predicted by the simulations were similar to those observed in the experiment. These are experiments done also with vesicles, and the fibrils here are attached to the vesicles. As I said, the model is simple and fast, which allowed us to investigate the details of the kinetics, which we did by network analysis. In this network picture, each circle is a microstate, and the connections between circles are those observed along molecular dynamic simulations. And the network picture reproduces the complexity of the fibril formation mechanism and also illustrates the polymorphism. So when we start with dissociated monomers, we observe the rather quickly formation of micellar intermediates, which then evolved either in the 2 protofilament protofibril or in a species with 3 protofilaments or in the mature fibril. And you can see here that there are traps or off-pathway mesostates that can accumulate during the process. So if one tries to calculate free energy barriers and nucleation barriers, which we did at that time, we saw that it was the nucleation barrier that was very influential for the population of the different fibril morphologies, essentially starting from the monomers, and after the formation of the micellar oligomers, there is a nucleus which can then evolve towards different protofibrillar or fibrillar states. And so, as of course observed in the experiments during the lag phase, so between the lag phase and the growth phase, there is the nucleus, which is the species that has 50% probability of going back, 50% probability of forming fibrillar states, and depending on the free energy barriers that are along the way, one obtains different protofibrillar aggregates. So this is now a two-dimensional picture, a schematic illustration of a potential free energy surface of aggregation. Along the y-axis here, I illustrated the number of aggregated molecules, and along the x-axis, one can schematize the number of protofibrils. So starting from the monomeric state, which is very floppy, the fibril formation can proceed along this pathway and generate one protofilament fibrils, or along this pathway, or along this pathway. Now, it happens more often than not that the process is under a kinetic control, because first a metastable state is reached, like in this case here, through multiple pathways. And from this metastable state, the process evolves then and reaches a kinetic trap, so a local minimum of the free energy. And because of this initial phase and the multiple pathways that lead to this nucleus that then evolves into a kinetic trap, other fibrils with more favorable free energy, like this one here, or this one here, which in this picture, it would be the global minimum of the free energy, they are not accessed. So it's really a kinetic control. So now, interestingly, computer simulations have predicted that fibril formation is governed by barriers and kinetic traps, so kinetic control, rather than the free energy of the final aggregates. Actually, there are atomistic simulations with an implicit model of the solvent by Martin Karpus and co-workers, which had provided evidence that dimerization is under kinetic control. And then our simulations with the coarse-grained model and 100 or more molecules. So the simulations have suggested that the diversity in the morphologies is a consequence of the variations in the number of protofilaments, which is what I have shown you earlier. And now these predictions have been confirmed afterwards by cryo-electron microscopy studies in Markus Fendrich group, published in 2019, that made use of brain tissues of Alzheimer's patients. So this is a picture from the paper of Fendrich's group. And you can see here the fibrils with different number of protofilaments, like this one here seems to have 2 protofilaments, this one here 3. And so this is, in my opinion, an interesting example where simulations made prediction of kinetic control and multiple possible species of polymorphism, and experiments later provided definitive evidence, actually from brain tissue of Alzheimer's patients. So because of this kinetic control, it is much more difficult to design drugs, so small molecules or antibodies that can modulate the process. And so one has to consider new types of drugs that have to modulate the kinetics rather than the end products of the self-assembly. And it is likely, or we and others have hypothesized that such drugs have to stabilize or could stabilize the fibrillar states that are not toxic. So this would be really the holy grail. And it's of course a rather unconventional strategy, because many studies have tried to develop small molecules or antibodies that hinder the fibril formation or fibril growth. Of course, the main question in this context is which of the metastable aggregates are innocuous, and actually we here I come to the second part of my presentation, in which we designed antiprion compounds in collaboration with the groups of Adriano Guzzi at the University of Zurich and Peter Nilsson in Sweden, who did the chemistry of the compounds I'm going to show you soon, as well as Beat Meyer and Anja Böckmann, who were working with solid-state nuclear magnetic resonance spectroscopy. Most of the work was actually done by Danji Wang, a postdoc in my group, who did the molecular dynamics simulations, and Uli Hermann, a PhD student or an MD PhD student in Adriano's group, who did all of the mice study. So the motivation when we started this project was that it is known that certain dyes interfere with the propagation of prion for instance, by stabilizing prion P aggregates in vitro and in vivo. This was evidence that Nilsson and Aguzzi had already gathered. And among the most interesting compounds, they were evaluating so-called LCPs, luminescent conjugated polythiophins, so polymeric molecules consisting of thiophene rings covalently linked and decorated with negatively charged carboxy groups. So there had evidence already 10 years ago that these LCPs increase the resistance of prion P scrapie to digestion by a proteinase that is used frequently to investigate the stability of prion protein scrapie. They reduce the prion infectivity, they reduce the amount of aggregated prion protein, and inhibit fibrinogenesis of recombinant prion protein. When we started, there was an NMR structural model of the fungal het S protein. So it's a fungal prion-like protein. This structure this structural model had been determined in the group of Beat Meyer, and the data allowed them also to dock Congo red, and they observed salt bridges between the sulfonate group of Congo red and the amino groups of lysine side chains in the het S amyloid structure. So Danji Wang made a very simple model of, or the simplest model possible of a prion-like structure, which is a beta sheet consisting of 3 peptides, threonine, alanine, K peptides, and carried out explicit solvent umbrella sampling simulation by pulling the polythiophene from the fibrillar model. And thanks to this very simple model, we could investigate its robustness. So again, this is explicit solvent simulations. The simplicity of the model lies in the very simplified structure of the prion-like fibril. And now the umbrella sampling explicit solvent simulations allowed us to calculate potential of mean force, so W, free energy, along the distance between the small molecule and the structure of the simplified model of the structure. And so this is the bound state here. This is the unbound state. And there was some robustness. For instance, this polythiophene in lysine, and the model consisting of serine, alanine, lysine, and the same for this other polythiophene. So we observed that there was some robustness in the choice of the peptides what was important was to have the lysine, and there are several lysines in the prime P sequence. So what happened next was that we used this free energy, so this potential of mean force calculations to screen compounds. The group of Nielsen had synthesized about 50 of these different polythiophins with different number of rings. This is a 4 ring, this is a 5 ring, and different decorations. This one has only 2 carboxy groups, this has 4 carboxy groups. And also the position of the carboxy groups was different, like in this case and in this case. So thanks to the molecular dynamic simulations, we could select a set of 7 or 8 polythiophins to be tested then in vivo experiments. And so here, Uli Hermann injected with a pump the compounds directly in the brain of mice. And we observed an interesting agreement between those compounds that had been predicted to have the most favorable free energy of binding, like Lin 50, 44, and 45, the green and orange profiles here. So they were consistent with the survival of the mice. So this is a plot of the that illustrates the in vivo experiments. So essentially here you have the time, you have the survival of the mice. There is a control at around, so the mice die at around 65 to 70 days. And these 2 compounds administered directly to the brain of the mice could prolong the life by 33 days or 56 days, which is substantial. These were control experiments, which somehow allowed us to see some good correlation between the in silico and the simulation studies and the in vivo results. So the simulations were useful to somehow restrict the number of experiments because the experiment is quite involved. One has to perform surgery on the skull of the mice, and there is a pump implantation. Then the prion protein are inoculated. Then one has to change the pump a couple of times, actually. And so because of the complexity of the experiment, it was very good that we could use simulations to somehow restrict the number of compounds to be tested. We, of course, also tried to see what happens with a more complex model of the prion protein. And so then we ran simulations on models of a segment of mouse prion protein from residues 177 to 216. Simulations without the LCP and with 3 copies of the LCP. And we always saw stabilization of the structural model in the presence of the polythiophins at both 310 Kelvin and 360 Kelvin. These 2 plots depict the distance of key side chain interactions like the ladder of arginines or the ladder of lysines here. And in the presence of the stabilizing molecule, the distances are smaller. So the fibril is more compact and most probably less frangible. I don't want to go into the details of the multidisciplinary approach that was used in this study. It involved, of course, chemical synthesis, in vivo experiments, solid state, nuclear magnetic resonance spectroscopy, and also the ND simulations of pooling, so calculation of the potential of mean force of different compounds to somehow reduce the number of compounds to be tested and generate a structure-activity relationship model. Right, then Z also tested the potential binding of LCPs to a model of the Alzheimer's A beta fibril, which consisted of octameric segment of 11 residues here. It's the sequence that includes a lysine, of course, because of the salt bridges with the carboxy group of the polythiophin, and it's close to the hydrophobic part of the A beta peptide. And here we also saw that the presence of the LCP augments the structural stability of the A beta parallel arrangement. Right, so we propose that, or we think therapeutically efficacious polythiophins reduce fungibility, so they stabilize innocuous aggregates and prevent fragmentation, secondary nucleation, and fibril extension. So we think that they act at this stage of the kinetics of fibril formation. So I'm already at the conclusions. We could confirm predictions of MD simulations by in vivo experiments. Conformation, we discovered a new lead compound, which was then tested also by intraperitoneal administration. One of these polythiophins extends the life of mice even upon intraperitoneal administration. And essentially, we did a structure-based design against protein aggregation. So the acknowledgements go to the main player, atomistic simulations of the importance of side chains in beta amyloid formation. Ricardo, who developed the phenomenological cost-scale model. Banji, who carried out the simulations of explicit solvent simulations of the polythiophins pulling out of a simplified model of the fibril, and Uli Hermann, who did all of the mice experiments. And the funding originates from the Swiss National Science Foundation, which has substantially funded my research group during the past 25 plus years, and funding also from the University of Zurich. And thank you for your attention.
Okay, thank you so much, Amadeo and Ioana. And now I think that if Rams agree, we can start the Q&A session.
Agree.
Okay, and I ask all the attendees that can raise their own hands, and they will be promoted as panelists, and they can pose questions directly to Amadeo and Ioana. In the meanwhile, that shy people raise their own hands. I have just a very naive question for Ioana. And the question is, we know that prion protein is a copper binding protein. And prion properties and toxicity and ability to spread and to promote fibril growth. Do you know, or are you planning to investigate how copper binding can modify the interaction of PRP with the POM1 or POM6?
Yeah, so but I don't quite remember what the outcome was. In any case, with these meta-alliance and simulations, it's quite difficult to do it because we need to parameterize the force fields if they're not well parameterized. I personally don't plan to go in this direction, so I want I focus more on the therapeutic side with these natural products. I don't know if Amadeo would like to continue in this direction.
Okay, thank you so much. And we have Astrid that can be promoted to the panel. Rams, can you do that? I don't know how to do that.
I tried. I don't think she wants to be on the panel. So she has posted the comment in the Q&A.
I don't see where is. Okay. Sorry, I didn't have to pop up the window. Okay. The question is, how those are the LCPS to patients trials? I think that is for Amadeo.
This is an important question. I think they're still far away. We do have evidence that they are not toxic, at least at the concentrations used for this kind of experiments. We do have evidence that they go, of course, to they pass the blood-brain barrier because they were efficacious upon intraperitoneal administration. But on the other hand, I mean, here there are two main issues. One is the prion disease, like Creutzfeldt-Jakob disease, for which there are very, very few patients. And the other is other types of much, much more frequent neurodegenerative diseases, like Alzheimer's or Parkinson. And I do not know what's the current status of the LCPs concerning clinical trials. Yeah.
Thank you very much. So it was very interesting lectures from both of you. I actually had one question also for Ioana. Can you hear me?
Yes.
Yes. So I tried to write it, but maybe it doesn't come up. So my question to you was, you had what I would call good and bad antibodies for your PRPC. So did you see any obvious significant differences in their interactions with the PRPC?
So the interactions are quite fine. So the way I was saying about the intramolecular interactions between the flexible tail and the globular domain, they are slightly different. Sorry, they are slightly different, but we cannot correlate this to anything.
Because that would be very interesting if you could correlate what's good and what's bad.
This is how we started. Yes, that would be the ideal case.
Yeah. Well, at least they do interact. That's good to know.
Well, yes, and they are slightly different, not significantly different, like not a lot different, but different.
Mm-hmm. But it was very clear from what you said at the beginning that one did increase the disease propensity, so to speak, and the other one decreased it, wasn't it?
Yes. Exactly.
Yes.
Yeah.
So you have
That's
to think more about what it could be. Yes. Thank you.
Yeah, we still need to explore. Thank you.
Yes, absolutely.
Okay, we can move to the next question from Alfonso De Simone. Excellent talk. I was wondering if using your cause-grained model, you can study the mechanism of secondary nucleation in comparison with primary nucleation.
Thank you, Alfonso. It's an important question. We have not done that. I presume we could. One would need to use more than 125 monomers. We did in the past simulations with pilot simulations up to 1,000 monomers. Nowadays, it should be even more feasible. Yes, why not? It would be very, very interesting to investigate fragmentation and secondary nucleation by this very simple phenomenological cause-grained model.
Thank you, Amadeo. We have another question from Lina Maria Peña. And the question is, you said that LCPS stabilizes in Nocus aggregates, but have you noticed something special about the structure of Nocus states compared to the ones that are not?
Yeah, thank you for this question. It is very, very difficult to investigate at atomic level of detail the structures of these aggregates. Some of them are also transient species. They are not very much soluble. So no, at the level of atomistic detail, no, I don't think we know that much. But there are, of course, a series of biochemical experiments in which they have seen differences, like differences with respect to proteinase K digestion and so forth.
Okay, we have another question from Carmelo La Rosa. Dear Amadeo, great talk. Your strategy to find a remedy for TSA goes in reverse to the one used by Biogen with its Alzheimer's drug. I think that Carmelo is referring to Aduhelm. Your work to stabilize fibrils, Biogen to digest fibrils. I have my own opinion on this. I would like yours. Okay. What is your opinion on?
Yeah, Carmelo, you ask a provocative question, and unfortunately I prefer not to answer. Also considering that antibody originates from the University of Zurich. It was originally developed by two colleagues of mine in the clinics. So I prefer not to answer this question.
Very diplomatic, Amadeo. Okay. We can move to John Straub. Question is, could you potentially fit your simulation data using the chemical master equation pioneered by Knowles and co-workers to gain insight into the mechanisms?
Yes, I think this is a very good suggestion. John, I think it was John Straub. Yeah, thank you, John. Yeah, it's a good suggestion. We have not done that, but I think in principle it's feasible.
Okay, we have another question from Shai. I will not try to read because it's not familiar. Shai Rahimpur, hopefully the right pronounce. Very interesting thoughts. Can you please give more detail about the cyclic peptide inhibitors? What are the mechanisms of action and whether they are selective?
Yeah, so this is a good question. So this is we're at the very start of this project, and we are designing the cyclic peptides ourselves. So we really rely on rational design, what we learned from the studies that I presented here, and also from the interface between the antibodies and the prion protein. The goal, so the ultimate goal, is that they are selective and they are highly potent. And we will strive to design them as such, and that's why we also hope to test them experimentally. And I mean, we have some ideas and tests, but I don't want to go into the details.
I don't see any further question in the box.
Can you ask?
I will check again, but
there are none, I think.
No other questions?
I have one question to Amadeo. Can you ask?
Sure.
Danilo?
Where is the?
No, I have a question to ask.
Okay, please, Rams.
Okay,
go.
great talks, Amadeo and Ioana. Amadeo, you showed a picture of the electron microscopic images where fiber getting attached to the membrane. And you also showed AMD simulation study showing that fibers can attach to lipid bilayer. Can you provide some insights into this, the factors that enable fibers to attach to the lipid bilayer? Is there any side chain or the peptide interacting with the lipid head group, or can you give some more details on that?
Yes, those results that I showed are based on simulations with a simplified model. The simplified model is a model of an amphipathic peptide. So we saw growth on the surface of a bilayer because of the amphipathic character of the coarse-grained model. I think for answering in detail your question, one would have to run simulations with atomistic models, and I think that there are lots of groups.
You saw,
studied
I'm familiar with the experimental results on this, but you saw the growth of fibers, or did you start with fiber structure in your simulation?
So we did simulations starting from the monomeric state and simulation starting from the fibril. And the interesting thing that we observed, at least with this simple model, is that the leakage from the vesicle, in the vesicle, we had probe particles, coarse-grained probe particles. The leakage we observed during the growth of the fibers.
Right, yeah.
Once the fibril was mature, so when we started simulation with the mature fibers, and the vesicle, there were interactions, but there was no leakage. So we postulated at that time that it's really the growth that damages the membrane.
Yeah, that's consistent with the experimental observation. Another question I have is, you mentioned about the micelles formation on the pathway to form fibers. But some of the amyloid peptides do form micelles, but they're off pathway. So the micelle formation need not be a step on the pathway all the time. So can you comment on this?
But yeah, with our model, we saw the micelles at the beginning. We saw also a coexistence of micelles and fibrils. So when we ran simulations with 1,000 monomers, we saw coexistence of micelles and fibril-like species. But I mean, depending on the beta propensity of our model, so our model had only one parameter, the beta propensity. There was more or less formation of micelles. But I think, well, of course, if the beta propensity was very low, then the micelles stayed there forever. And because the micelle in the model don't have any beta sheet-like aggregates, and so if the model had very low beta propensity, then the micelle stayed forever.
So the coexistence means they don't have to be, they're not in equilibrium, right? Are they in equilibrium, fibers and micelles?
Well, they are in equilibrium on a short time scale, because then eventually they evolve to equilibrium monomer fibrils. At least in our model, the final equilibrium was monomer fibrils.
Okay. Thank you very much.
Welcome.
Amadeo, another question.
Sure.
In your simulation, when you study the ground of the fibril on the membrane surface using vesicles, the radii of the vesicle is short or large? Because the radii vesicles there is very important in the kinetic of fibril ground.
Yes, I mean this remark is very important, and the curvature was quite high. So the vesicle was quite small because of the required simulation time. I don't think we did simulations with a very large vesicle, which would have had a lower curvature. I mean, these simulations were done more than 10 years ago. So nowadays one could do much more, of course. And so in principle, nowadays we could also investigate what you suggest, which is the difference between vesicles with few particles and vesicles with more lipids. So the influence of the curvature.
Okay, thanks.
Yeah, so thank you guys for the great talks. It was really enjoyable, and again, thank you for coming to the ZOOMinar series.
Thank you
and,
for having us.
Kopo, see you in the audience. Come. And,
Thank you for
getting
this
some
opportunity.
Thank
Yeah,
you very
Very
much.
much,
Bye.
very
Danilo.
interesting.
Yeah, you're
Lectures.
welcome. Yeah.
Danilo,
Ciao,
thank you.
Amedeo.
Bye.
Ciao,
Bye.
Amedeo. Bye.
Bye.
Thank you.
Bye bye.
Bye.
Thank you. Bye.
Bye.